The emerging field of pharmacogenomics, the study of how genetic variation influences drug and supplement response, offers one of the most intellectually honest frameworks for thinking about individual differences in ND plus precursor response. Not everyone metabolizes niacin, NR, or NMN the same way. Genetic variants in key enzymes along the ND plus synthesis and clearance pathways mean that what works remarkably well for one person may produce minimal response in another. Understanding this genetic dimension doesn't just explain the variability in supplementation outcomes, it potentially allows for a more targeted, personalized approach. The NMT enzyme is central to this story. NMT, nicotinamide N-methyltransferase, methylates nicotinamide to produce N-methylnicotinamide for urinary excretion, effectively clearing NM from the system. Genetic variants that create high NMT activity mean faster NM clearance, which may reduce the sirtuin-inhibiting NM accumulation discussed earlier. Individuals with high NMT activity might theoretically require higher doses of any ND plus precursor to maintain circulating levels, since the pathway is clearing metabolites more aggressively. They might also benefit from additional methylation support, supplementing trimethylglycine, SAMe, B12, and folate to sustain the methylation reactions that NMT depends on. NAMPT variants affect the salvage pathway efficiency that all cells use to recycle nicotinamide back into NAD+. Individuals with compromised NMPT function have reduced ability to recycle NM into NAD+ through the primary salvage route. For these individuals, there's a theoretical argument that NR or NMN, which can bypass certain steps in the salvage pathway, might be more effective than niacin, which relies more heavily on the price handler pathway and efficient salvage recycling. Whether this theoretical advantage survives actual human measurement in compromised NMPT individuals has not been systematically studied. NPRT, nicotinic acid phosphorybosyltransferase transferase, is the enzyme that specifically processes nicotinic acid, the flushing form of niacin, through the price handler pathway. Variants affecting NPRT activity directly affect how efficiently niacin, specifically nicotinic acid, is converted to NAD+. Poor NPRT function could theoretically reduce niacin responsiveness while leaving NR or NMN-dependent pathways intact. Accessing this genetic information is relatively straightforward using consumer genomics services. 23andMe or Ancestry DNA raw data can be uploaded to third-party interpretation tools to identify relevant SNPs in NNMT, NMPT, and NAPRT. The important caveat is that genetic insights in this domain are probabilistic, not deterministic. A variant associated with lower NMPT efficiency doesn't guarantee that NMN will work better for you. It raises a hypothesis worth testing with personal biomarker data. Genetic information should frame the experiment, not replace it. The methylation connection also raises a practical safety consideration. High-dose ND plus supplementation in individuals with poor methylation capacity could exacerbate methylation depletion, raising homocysteine and potentially increasing cardiovascular and neurological risk. Monitoring homocysteine levels and ensuring methylation cofactor adequacy is a responsible component of any serious long-term NAD+ protocol.